So this is our presentation about the challenges of biohybrid and bioinspired robotics. First, we're going to talk about the background of this side of robotics and its importance in the national scale, talk about the challenges the biohybrid robotic faces, and then propose a solution for one of those challenges.
The first background is human-like perception. Human-like perception is the ability for a machine or system to carry out tasks automatically without human input. The machine or system must learn from its environment and adapt to changes accordingly. And knowledge bases, most of the time, a knowledge base is required to assist in data storage and analysis. Here's an example of human-like perception implemented. Is the Tesla's fully autonomous vehicle, which uses computer vision. Computer vision is the use of images, videos, GPS, sensory data to create a detailed, active picture of the environment. The Tesla then uses this image to adapt to scenarios like road collision in real time. In some cases, the vehicle can predict a vehicle collision before it has happened and then adjust its speed or direction in order to prevent harm to the occupants. We also have the assembly line. Assembly line robotics can, for example, in vehicle manufacturing, can be fully articulated robots that spot weld parts onto vehicles. However, human-like perception is still in development and is not perfect. For example, the Tesla is not fully autonomous at the moment and still requires human input. Here we have an example of human-like perception implemented in a real-life scenario. This is called the Roomba vacuum. It uses computer vision, so images, cameras, and sensors in order to vacuum the whole room and can detect dirt and go to the dirt and vacuum it up.
So we're going to talk about the bioactuation history. In 2009, the first insect dorsal vessel or heart tissue manipulator was introduced. It was used because of its environmental robustness and low maintenance cost. Existing bioactuators at the time could not work outside of culture, and this limitation in the environment was a difficult problem to solve. Dr. Tayama and his group proposed to package the tissue with its medium, and this way we could get an actuator that could work in air and outside of culture. In 2014, a long PDMS skeleton with a rigid head and a compliant tail is used to create the structure for the cells to grow on. The cells would contract and bend the tail to propel the robot forward through electrical stimulation. However, due to its scale, it could only operate under a low Reynolds number environment and in culture medium. In 2014, one of the first uses of optogenetic C2C12 cells was revealed. The modular skeletal muscle-powered bioactuator was able to generate 300 micronewtons of active tension force in response to non-invasive optical stimulation. The use of optogenetics meant that there is less danger for the cells to be damaged due to electrical stimulus. Lastly, in 2016, a group of scientists was able to create a biohybrid batoid that used optogenetic C2C12 muscle cells as well. The cells formed myofibers that bound tightly to the skeleton of the elastomeric body of 16.3 millimeters in length. Those myofibers produced the necessary force to produce the batoid robot, to propel the batoid robot up and down, and this was the first demonstration on how we can control a robot using bioactuators accurately and remotely.
A self-healing material is a cutting-edge approach that aims the increase of independence of an autonomous robot. Self-healing elastomers can heal microscopic and macroscopic damage and can be implemented to soft robots that face tasks in which shocks and impacts are common. The technology is still in development, and this image shows the project of Terry et al. That designed prototypes of self-healing actuators for soft robots that demand resistance. In the project, macroscopic damage could be healed entirely using a mild heat treatment, and the full performance of the actuators was nearly completely recovered after healing. The main objective of self-healing is to achieve mechanical robustness and rapid self-healing properties, and the advantage is to increase the operation time and the reduction of repairment costs.
Now we're going to talk about the research gaps that bioactuators have. One is scalability. The largest bioactuators was used in the robotic ray as mentioned before. Its actuators consisted of 200,000 live cardiomyocytes. However, even with this great achievement, it's still a long time until we can grow as much muscle cells in our arms and produce the equivalent force. Another is control, as we still don't understand how to control specific muscle groups or how to produce the specific forces. We are limited only to when they contract. Some other ways of stimulation were proposed through chemical or magnetic control. However, they proved extremely costly and inaccurate. Lastly, we still can't recreate muscle-like properties. Promising developments have been seen, especially with the muscle rings in 2014. Through constant training by stimulation, the tension forces produced by the muscles increased by a factor of 5. However, there are yet to be developments on how well the bioactuators can heal without changing their original characteristics.
And the project's aim is to achieve more energetic autonomy, efficiency, and increase the independence of the biohybrid actuator to external stimuli. And optogenetics, a dielectric elastomer actuator, a microfluid logic, and/or piezoelectric actuator approach could be adopted to achieve the objective. The achievements of the research would be done through the implementation of advanced mathematical models, optimization algorithms, and software tools for optimizing the design and for predicting the performance of future biohybrid systems.
National importance. If successfully developed and implemented, could lead to a new generation of actuators. This will lead to great advancements in the medical sector. We could have self-based actuators with prosthetics that could self-heal and actuate at command. We could also have robots that could self-heal and provide their own energy, making them a part of a sustainable ecosystem. So the first proposed solution is the DEA. DEA devices simply convert electrical energy into mechanical energy and are also reversible. A simple example of a DEA would be made up of a thin elastomer membrane, also called a polymer film, that is placed between two stretchable electrodes. To activate the actuator, a voltage difference is applied to the electrodes. The difference of charge between the two electrodes leads to electric fields being induced in the membrane, which cause attractive electrostatic forces between the electrodes. These attractive electrostatic forces arising between the electrodes cause a mechanical compressive force in the form of electrostatic pressure to be exerted on the membrane. Due to the elastomer's resistance to compression, the decrease in thickness caused by the compression causes lateral expansion. Researchers in the Volume 16 lab on a chip created a DEA-based deformable bioreactor. The researchers, to demonstrate a proof of concept, cycled a population of lymphatic endothelial cells (LECs) with a uniaxial strain between 0% and 10% at 0.1 Hz during a 24-hour period. They found that this caused stretch-induced alignment along with elongation of the LECs. We can see this in the image over here. We have the control cells, which are just non-stretched, and you have the simulated cells where you can see elongation and stretching. With experimenting, the researchers found that the DEA-based deformable bioreactor, which is to the left, could generate up to 35% uniaxial tensile strain on cells. This provided the first demonstration that DEAs could be integrated with living cells and used to control mechanical aspects of these cells. Although this application of DEAs in cell cultures is in its infancy, it is very promising, and if scaled and developed correctly, could lead to a new generation of bioactuators. Such bioactuators will be capable of producing their own energy and be able to self-repair.
To show that optogenetics could also be a good alternative to DEAs, we can look at a study that was done in 2014, where the contractile forces and the response times was compared between photo and electrical stimulation. To get the photo-stimulated results, a blue LED was used to stimulate a genetically modified C2C12 cell that expressed CHR2 genes. By using a blue light, we ensured that more energy could be absorbed by the muscle cells, leading to a stronger contractile force. Both types of the cells were stimulated for 100 millisecond periods at different frequencies, and as we can see from the graphs, the results are relatively similar for frequencies of 4 Hz, and slight differences can be seen between the two in frequencies reaching 5 Hz. This means that photostimulation may be a better alternative as we reduce the risk of damaging the cells due to electrostimulation. Some of the problems seen during the experiment was that some of the cells weren't able to differentiate fully to muscle cells. This meant that the forces produced by the cells were weaker, and less cells could be stimulated by the light. More study needs to be done in order to increase the consistency of these results. Another is that the cell would degenerate after a few hours of use. We propose a solution to this, which is using insect DVT instead of mammalian cells due to the robustness, proven by its ability to retain 80% of its strength continually for 30 days without the need to replace its medium. Some of the promising results was seen in the muscle rings in 2014, which is a way to control the forces produced by the actuators by adding more or less of the muscle rings. Another good thing that came from the study was to show that we could replicate the muscle's ability to get stronger through training. Through constant stimulation of 1 hertz for 12 days, the muscle rings was able to increase its force from 195 micronewtons to 283 micronewtons. Therefore, photogenetics may be a great alternative to DEAs as well.
Microfluid logic is a cutting-edge technology that can be used to provide independence to robots. Microfluidics is the science responsible for manipulating fluids that range from 10 to the power of -9 to 10 to the power of -18 liters, and is being applied in microelectronics using elastomeric materials. The Octobot, as shown in the image, is a pioneer and cutting-edge example of an entirely soft and autonomous Bioinspired robot. With the adoption of a pneumatic-based microfluid logic, the robot can move autonomously for 4 to 8 minutes due to combustion reactions of two fuels. The Octobot could work for 8 minutes autonomously, but the objective is still to increase operation time of actuators due to not be feasible to implement it in rescue activities or another kind of tasks in real life. So the study and implementation of more complex microfluidic circuits for a Bioinspired robot can be an approach to achieve a higher autonomy time. This research can be done with computational fluid dynamics analysis, such as ANSYS or COMSOL, to understand better about the features of the flow, and we can do validations with experimental approach to compare both. A PID controller simulation could be adopted to decrease the error, increase the autonomy, and adopt or more complex logic gates could be another approach. Piezoelectric actuator is designed for micrometer scale robots in the aerial field and is an alternative to reach external stimuli independence. As suggested by Jeffers et al. In 2019, solar cells can be used as the source to achieve autonomy to microrobots. So the solar cells still needs to get more efficiency to improve the efficiency, and this can be done with another kind of simulations involving computational fluid dynamic analysis using a heat transfer approach and a validation with experimental results. More important than that is to implement a PID controller to the wings. Due to the wings currently provides a behavior of open loop control that is uncontrolled. Besides that, it's important to implement multi-layer actuators and blocking diodes to provide and improve energy recovery.
Just to conclude, overall the best approach for a possible solution is solely dependent on the development of such technology. All 4 proposed solutions, however, are in their infancy and require much more research and development until they can be fully implemented into a working application. However, as highlighted in the 4 solutions, if correctly scaled, developed, all 4 solutions could be promising candidates for bioactuators and lead to a new generation of actuators for the field of robotics. Thank you for your time.
Thank you.